Thank you so much for your great introduction. It's my great honor to have you all here. I also would like to thank Brick for inviting me to present this my recent work during this webinar. And my voice is kind of funky right now because I've caught a real bad cold. So if you can please excuse my weird voice, I would be much appreciative. So with that said, I'm gonna get started. So this is about breast cancer subtype transition from very malignant breast cancer subtype called basal-like breast cancer subtype into more of less invasive breast cancer subtype called luminal A. So I'll go through the presentation slides by this order. So first of all, I'm gonna be explaining about the background research goal and what kind of data sets that I use for this particular study. And then how I constructed network model and what kind of network control analysis that I performed to find potential targets for such transition, and how I validated the identified targets from the network analysis. And then I'm gonna wrap up this presentation with the summary. So breast cancer has the highest cancer-related mortality and highest incidence rate among women worldwide. And this can be divided into 4 major subtypes called basal-like or triple-negative, luminal A, luminal B, and HER2-enriched subtypes. And other than triple-negative breast cancer subtypes, they can be usually treated with hormonal therapy, or there are targeted therapy available for those subtypes, whereas basal-like or triple-negative breast cancer subtype doesn't really have specific therapy for them, so they usually have to rely on chemotherapy for treatment. On the other hand, luminal A breast cancer subtype occurs in the most differentiated state during mammary stem cell development. So if you look at this hierarchy over here, it starts from mammary stem cell differentiating into bipotent progenitor cells, and then onto these more of differentiated version of the cells. And tumor genesis can happen at any of these stages. So if tumor genesis happens more of during the earlier stage of the development, like over here, it's going to be called basal-like or triple-negative. And if the tumor genesis happens at the very later stage, like from here, it's going to be called luminal A. And also luminal A, there are more than about 70% of breast cancer patients belong to this luminal A group. And they are usually treatable with hormonal therapy called anti-estrogen receptor alpha therapy. So there is treatment available for those patients. And if you also look at this survival here, basal-like breast cancer or triple-negative breast cancer subtype has the worst prognosis, whereas luminal A has the best prognosis of these breast cancer patients. So there have been numerous studies that are suggesting potential, therapeutic targets or possible cure for these malignant breast cancer patients, but there was no significant effect so far. So for this particular study, we took a slightly different approach to solve this problem and identify, genes that can sort of make this malignant tumor cell type transdifferentiated into more of less invasive subtype called luminal A cancer cells. So the anti-ER therapy that are usually valid for these patients can also be an alternative treatment option for these basal-like patients as well. So this is an overview of this study. So we first constructed network model specific to basal-like and luminal A breast cancer patients, and we validate the model. And then identify targets using network control strategy, and validate those identified targets using in silico simulation as well as in vitro experiments. These are data sets that I used for this particular study. We used KEGG, String, Trust, Literum for public databases and literature to kind of reference those interactions within the network model. And we also use patient data sets and cell line data sets to kind of validate our analysis. So I'm going to be explaining about how I constructed network model. So prior to network construction, we used the ER and EGFR signaling pathways, which are known to have most influence on cell growth and drug response from basal-like and luminal A breast cancer patients. And we use these signaling pathways as the backbone structure of our network model. And then we performed differentially expressed gene analysis, DG analysis, using TCGA datasets of basal-like and luminal A patients to find marker genes specific to luminal A and basal-like patients. Among top 10% ranked genes, which are shown here, we selected genes that are highly expressed in one subtype, but lowly expressed from the other based on their p-value. And we confirmed that these selected genes are highly expressed from basal-like or lowly expressed on the other luminal A breast cancer patients, and vice versa for the other genes as well for luminal A DGs. And we confirmed the results using TCGA datasets, metabolic patient datasets, as well as the CCLE breast cancer cell line datasets. So this is our final network model we constructed. This model has 30 nodes in total and 73 links, interaction links. And based on these genes, we also analyzed their copy number alteration, mRNA expression, and functional mutation profiles of these genes from 21 basal-like breast cancer cell lines. We did this analysis to construct this functional genomic profile that kind of encapsulates every bit of CNA, mRNA expression, and mutation profile of these genes within the network model. And then we use this functional genomic profile to reconstruct our original model into cell line specific model to find specific targets from the cell lines. So for this, we use this information to kind of cluster the cell lines to see which cell lines can represent the overall breast cancer, basal-like breast cancer patients or cell lines. And you can see that these are kind of clustered into two major groups, and we selected one cell line from each cluster to kind of represent basal-like breast cancer cell lines. And then we selected MDA and B231 cell line and BT20 cell line to represent basal-like breast cancer cell lines. And these are the cell line specific models that we have. And then after the construction of our network model, we use Boolean network modeling. Boolean modeling suggests that the value of each variable is 0 or 1. So it's binomial, and the change or activity of these each node's variable is controlled by a Boolean function called OR gate, NOT, or AND gates. So this is very useful modeling method in systems biology because we don't really need detailed kinetic parameters for analysis. So this Boolean network modeling is very widely used in systems biology and the research associated with it. And these are kind of reference for the interactions that we included in our network model. So these are references that shows the interaction between the nodes within our network model. For validating, after construction of our network model, we had to validate whether our network model is biologically valid. So for the validation, we try to mimic the actual biological phenomena using this network model. Since within our network model, there are two major signaling pathways, ER and EGFR signaling pathways. So we wanted to validate whether these signaling pathway is working properly as the ligand binds to their receptor. So we use estrogen and EGF as our ligands, and we manipulated these ligand intensity to see whether the corresponding signaling pathway is working properly. So we kind of mimic the situation where, if we increase the intensity of estrogen here, it's going to bind to the ER here, and then it's going to activate the downstream molecules. And for the same case for EGF, when we increase the intensity of EGF from 0 to 100%, EGF and its downstream molecules are increasing their activity according to the ligands that it's binding to. So we can see that from this panel, it's working properly. And going a little further, in real biological phenomena, there's no such thing as single ligand binding to one signaling pathway. So we wanted to kind of mimic when these two ligands are simultaneously binding to the ligands within the network model. To mimic this situation, we manipulated the intensity of these ligands differently. So we gradually increased the intensity of EGF from 0 to 100%. And at the same time, we kind of fixed the intensity of estrogen at different levels from 0 to 50% and 100%. And we look at the patterns of these downstream molecules, and we were able to see that this can kind of mimic the situation where basal-like cells are kind of more dependent on EGF rather than ER, and these patterns are showing exactly that pattern. So we were able to see that our network model is valid and can mimic the situation in basal-like cells as well as luminal A cells where it's responding to estrogen receptor ligand, which is estrogen. So now I'm going to go through how we identified our target genes from our network model. So we use this network control strategy called logical domain of influence, or LDOI, and we also use expanded network to unravel the mechanism of basal-like to luminal A transition in our Boolean network model. So first of all, expanded network is like a trajectory of every possible state within our network model by integrating its Boolean logic information. So moving on from one node, so for example, moving on from one node to another, these interactions and their relationship is kind of projected onto this expanded network model here. So every single step of these interactions and their molecular interactions are kind of projected over here, and it shows their logical relationship. So for example, if you look at this expanded network here, and particularly for node called 4, so N4 means node number 4. And if you look at this, if you want to turn off N4, these N0 and N1, both of them had to be turned off from this situation over here. And also, if you want to turn on N4, you need either N0 or N1 to be turned on, which is shown here. So these logical relationship is kind of projected on expanded network over here, and we can also use this to, what kind of mechanism behind the scene it's for this particular transition. So by using this control theory, we search for what genes or nodes and how these should be manipulated in order to have our desired state, which is luminal A state. So we look for, we put in our desired state, which is to turn on ER alpha and luminal A DEGs over here. And at the same time, turn off EGFR and its downstream molecule, AKT and ERK1 and 2. And we were able to find that BCL11A and HTAG1 and 2 should be turned off from our original network model. And we also used our cell line specific network model, BT20 and MDA231, to see, what combination would be kind of overlapping between each other. So we were able to see that there were, several options, combinatorial targets that we found from these networks, but only BCL11A and HTAG1 and 2 were kind of overlapping from all of these network model. So we figured that, BCL11A and HTAG1 and 2 inhibition of those genes would be the ultimate target for our network model. And we also look at what kind of mechanism behind that scene after inhibiting HTAG1 and 2 and BCL11A. So we use this expanded network model here to find what happens after inhibiting BCL11A and HTAG1 and 2. And we kind of organize this trajectory into this transition map over here. And after inhibiting HTAG1 and 2, it's kind of turning on ER alpha signaling pathway and luminal ADGs here, but it's at the same time turning on the EGFR signaling pathway and its downstream molecules here. So, as some of you might know, that HTAG1 and 2 is histone acetylase complex 1 and 2, which are known as epigenetic regulators. So these are not just turning on the luminal ADGs and its related genes, it's also turning on EGFR and its downstream molecules and the basal-like DGs as well. So it's, kind of expected. So as you can see from this mechanism, that HTAG1 and 2, inhibition of HTAG1 and 2 only is not sufficient to make the transition from basal to luminal A. So we also look at this transition over here, what happens after inhibiting BCL11A. And as you can see, after inhibiting BCL11A, EGFR and its basal-like related genes, including EMT markers such as ZEP1 and SNAIL here, are also turned off after inhibiting BCL11A. So we can see from this transition and the mechanism that both BCL11A and HTAG1 and 2 should be inhibited to sufficiently induce basal to luminal transition in our network model. So after identifying these target genes, we wanted to validate whether these genes are valid and actually can induce the transition. So first of all, we use Boolean perturbation analysis for computational analysis. So we used attractor stimulation and manipulated these genes from our network model and measure the basin size or percentage of luminal A phenotypic states within our network model. So these are attractor states, showing some of them, like from here, actually all of them from here are considered to be basal-like phenotypic states, whereas after inhibiting BCL11A or HTAG1 and 2, it's kind of making a transition slightly. So about 30 to 50% of the states are kind of luminal, whereas the rest is still basal. And after inhibiting both BCL11A and HTAG1 and 2, it's can induce the basal to luminal transition for 100%. And the result is consistent using BT20 cell line specific network model. And this is kind of showing the actual percentage of those basin size or percentage of basal or luminal A phenotypic states. Similarly, so before inhibiting anything, it's 100% basal-like phenotypic states, whereas inhibiting either BCL11A or HTAG1 and 2 can only induce, about 34 to 50% of basal-like phenotypic states remaining, whereas after inhibiting both BCL11A and HDAC1 and 2 can induce 100% luminal A states within our network model. And the results was consistent for BT20 specific cell line network. So after confirming our results using in silico simulation, we further validate the results using in vitro experiments. So we use shRNA and the drug called romidepsin that specifically inhibits HDAC1 and 2. We use those to inhibit both target genes from our cells, and then see whether the transition happens by measuring RNA expressions of ESR RNA, ESR gene, and protein level of ER alpha, which is phenotypic marker for luminal. And we also measure the activity of EGFR and its downstream molecules. And as you can see, after inhibiting both BCL11A and HDAC1 and 2, the results, the mRNA expression of ESR what was increased up to 10-fold. This is two different sequences. So this one shows about 10-fold increase, and this one is showing about like 11-fold. And from BT cell lines, the results was consistent. It shows up to 4-fold of ESR1 gene expression increased. We also measure the protein expression of ER alpha. So this is ESR is ESR is gene for ER alpha. So we measure the protein level of ER alpha, and it shows the increase of ER alpha expression after inhibiting both BCL11A and HDAC1 and 2. And simultaneously, the activity of EGFR and its downstream, ERK, is also decreased after inhibiting both BCL11A and HDAC1 and 2, up to 100-fold for this particular cell line. So we were kind of able to prove that the phenotypic marker for luminal A cells has been increased after inhibiting those target genes, whereas the phenotypic markers for basal-like cells have been decreased. And we also measure the drug sensitivity of those cells after inhibiting both HDAC1 and 2 and BCL11A. And after the inhibition, you can see that it became more sensitive to tamoxifen here and here from both cell lines. And morphologically, you can see that the cells became more sensitive to tamoxifen after inhibiting both BCL11A and HDAC1 and 2. So from this, we can hypothesize that overexpression of these genes in luminal A can, will result in contrasting results, that luminal A cells would become more of basal-like and become resistant to tamoxifen. So we overexpress BCL11A and HDAC1 and 2, these genes in luminal A cells, and observe the results. And we can see that ER alpha expression, as expected, decreased, whereas the activity of EGFR and ERK increased after overexpressing these genes in luminal A cells, and cells became, resistant to the drug, whereas the control cells, luminal A cells, were very sensitive to the drug, as shown here. We also look at the patient survival based on these gene expressions in patients using TCGA and metabolic patient data sets. And we were able to see that high expression of BCL11A, HDAC1 and 2, and HDAC2 is related to patient survival, and high expression of these genes can result in worse prognosis than that of patients with low expression of these genes. So this is patient survival from basal-like patients. This is for luminal A patients and in combination. And we also confirmed the results using metabolic data sets, and it's shown here, same thing. high expression of these genes will bring worse prognosis or worse outcome in patient survival. Finally, we use geo-patient data sets to measure tamoxifen sensitivity based on their expression levels. So we first categorize the patients into 4 different groups. So BCL11A, HDAC1 and 2 high group, and BCL11A low group, HDAC1 and 2 low group, and both of these genes in low expression in patients. And as expected, we were able to see that these genes with with the expression of these genes lowly expressed, the patients were more sensitive to this drug, tamoxifen, whereas high expression of these genes within the patients can cause very resistant to the drug, which is shown here. And we also perform GSVA analysis to kind of compare the gene expression levels of these genes, luminal A genes and basal-like genes. And as you can see, this BCL11A HDAC1 and 2 high group is, kind of more enriched in basal high and luminal A gene expression patterns, whereas these genes in low expression in patients were more enriched in luminal high and ER alpha related genes, which is shown here. So for summary, we constructed our own network model that are specific to basal-like and luminal A patients. And we found the target genes that can sort of make the transition, transdifferentiate basal-like cells into luminal A cells in our network model. And we identify the target genes using systems biological approach called LDOI, and we validate the results, computational simulation results using in vitro experiments for this particular study. So this is the result, and thank you so much for your attention. And if you have any questions, please ask me now.